It is now my pleasure to welcome to the floor Panna Sharma, President, CEO, and board member of Lantern Pharma, who will be speaking on transforming oncology drug development by leveraging AI, including real-world examples from an emerging clinical stage biopharma company. Panna, thank you so much for joining us today. It's a pleasure to have you with us, and the floor is yours.
Franco, thank you very much. I'm going to be talking a little bit about what we're doing at Lantern. We're one of the companies that I think Deep Pharma and Andre mentioned earlier, where we're really changing and transforming the pace and cost of drug development by using a platform-driven approach. I'll talk a little bit about the company so people have context, but then dive into how specifically we're using AI to enhance and automate various aspects of our drug development decision-making. So Lantern Pharma, we're a publicly traded company. We've raised a little over $100 million since I joined the company 3 years ago. Our ticker is LTRN, and we may or may not update these statements I make today. So I think we're all familiar with the big megatrends that are available. We believe that these megatrends, as our last speaker pointed out, are changing the ability to generate new medicines faster and cheaper, and also with more precision. And a lot of that is that the data sets that are coming out now are higher quality, they're more readily available, and you can use them to train different ways to create correlations. And at the same time, the rise of sequencing and biomarker studies globally, it's becoming cheaper and faster. And ultimately, executives and entrepreneurs are being rewarded for taking these kinds of brave new business models. So we're at the forefront of applying many of these megatrends specifically to cancer. Cancer is a big area, $2 to $3 billion to make a drug, a decade, lots of data, lots of trials. So some interesting data sets out there that can be used to do the training. We were featured in Fortune magazine about 2 years back as one of the companies reshaping medicine through machines. In pharma, there's a big need for it. As many of you know, ROI in internally developed programs is low, and the trials that use biomarkers are on an average of 4 to 5 times much more successful. So we're taking the fundamentals of this business model. We developed our own in-house AI platform called RADR. We use RADR to assemble tons of genomic, clinical, and response data to help us clarify mechanisms of action, help us understand responder groups better, and potentially either rescue drugs that didn't quite meet their endpoints or develop entirely new drugs. And our business model is a little bit of both. We have both rescued agents now that are in phase 2, as well as totally new de novo drugs that are now in IND enabling studies or in phase 1. So our RADR platform is now about 11 billion data points. That's a massive increase since last year, about 10x increase when we reached a little over 1 billion last November. And it allows us to do 4 very important things. It allows us to accelerate the drug development timelines by taking what can take years or months and shortening that to weeks. It allows us to uncover new therapeutic opportunities where we can exploit the mechanisms that we're understanding better. It gives us insights into how we can create combination therapy programs. And very importantly, it allows us to expand our ability to collaborate with other partners, since many of the programs can be done with other drugs. These are all great ways to add value to an emerging biopharma company like ours. Now, we focus purely only on oncology. And so RADR at the core of it is an integrated data analytics and machine learning platform. We've automated various aspects of the data analytics and of the machine learning. We're using cutting edge machine learning approaches where we've automated the learning across lots of different algorithms. And more importantly, we've allowed now to generate hypotheses quickly, and we can validate those hypotheses in the wet lab or in animals and ultimately in humans. And we're scaling this platform and expanding it. And we believe oncology is a perfect area to apply this set of problems and this approach first. So how do we currently use it? As I mentioned, there are 4 big use cases. Understanding the mechanism of action, this is central to oncology drug development. Deriving ML-based signatures, which is critical because these signatures give us insight and they move things from being purely black box based to now having an understanding of what those very specific signatures are doing. What's the interaction between the drug and the cells, or the cells and the drug and the other factors. It allows us to identify drug combinations faster, which drugs will work synergistically or additively or probably against one another. So there's no reason for that. We can then look at drug classes, drug types. We can look at combination approaches, and we've identified several of those already, many which are under patent, and many we've actually now entered into trials with. And ultimately, it also allows us to identify new indications. I'll try to give use cases as much as I can with the amount of time that we have today. And I'll also, of course, try to leave time for Q&A because that's always very important. As I mentioned, one of the most important things is really the number of data points. And this is very central because without data, that's the lifeblood, I think, as one of the speakers said earlier. It allows us to have a more expansive set of cancers that we can look at and gives us more power and precision to the algorithms. And also we can train algorithms to do various things. And this AI platform, as it grows, it allows us to focus on more combinations, on rarer cancers, and also improving how the algorithms work, from everything from cell lines to PDX models to animals to actually real patient data. Over 80% of the data in our platform today is from real world patient data, and the other 20% is from cell line and PDX, approximately. One of our collaborations actually is with Actuate. We're looking to do more collaborations. They have a best-in-class GSK3 beta inhibitor, where they were looking to identify two very important things. What is the biomarkers that can help us predict the response in patients? And very importantly, what are some new indications? And GSK3 beta inhibitors are a very promiscuous type of drug. They can work in a lot of different areas. So pinpointing what is the most important area and the most likely to succeed in the clinic is a multi-$100 million question. And so when you can add data to this, it gives them better insight. But we're not doing it really for others. We're not much of a service provider. We believe that in kind of eating our own dog food. So we're really applying it to our own portfolio. When we went public last year, we had 3 drugs programs in development, one in prostate cancer, one in non-small cell lung cancer, and one in glioblastoma. Today, we have 8. We launched pancreatic cancer, ATRT, which is another CNS cancer. We launched an indication in blood cancers, and also bladder and hematologic cancer with a totally new drug, and also a combination that morphed more into an antibody drug conjugate that was predicted to have tremendous efficacy in certain solid tumors. And that's our antibody drug conjugate program, LPA18. So that's a vast amount of growth in about 15 months. And again, we try to patent all of these ideas, everything from composition of matter to combination to methods of use to signatures to even fundamentals of the AI platform itself. So we've got 90 patents issued and pending across 14 patent families, and that's growing. We filed over 15 patents this year alone. So let's get into the architecture of RADR. We have various sources of data, our own curated data that we get from DepMap and CellMiner and TCGA and CCLE. There's publication data repositories, and then our own proprietary drug studies and those from our collaborators like Fox Chase and Georgetown and Johns Hopkins. As the data gets ingested, we always grade the data, we curate it, and we put it into our data lake. And then we are able to then process the data for biomarker discovery, various insights that are important for drug development, and then a layer of microservices on top of that. So let's talk about, now I think our data points are over 11 billion, close to 100,000 different samples from a vast range of oncology specific data sources that are all then curated and cleaned up. They go across a lot of different omic areas, and many of these we will be continuing to grow. As I mentioned in a press release earlier, we'll be growing a lot of immuno-oncology data in the next 12 months. And that'll be a big area of focus as we get more into the immuno-oncology space. So our sample types, again, we focused heavily in the last campaign of growth in brain and blood cancers. So that's a big area for us. Obviously, prostate and bladder are GU cancers that we have in development, and that's a big area. And we also have a phase 2 trial in non-small cell lung cancer. So as you can imagine, that's also been an area of a high number of samples. So as we get more data, we'll have a broader set of cancers, broader cancer subtypes, and including in our next big campaign, lots of rare cancers. And so again, we're looking for collaborations in this area. Our data lake allows us to look at various sequence types, data sources, publication dates, and so that we can get smart about what data we pull in to do our work. So once we have all this data, it sits in various data lakes at S3. We do all our programming and version control in Jupyter and GitHub and R or Python. You know, we're pretty independent, but all of that is containerized in what we call CodeOcean. Our machine learning is all done in MLflow and Nextflow. We'll talk a little bit about that. And we've automated a lot of the reporting. We're beginning to automate a lot of that so that we can get insights that we can share across the team and with our collaborators to do the work. We have a collaboration with the NCI. We're one of the few companies that have a collaboration. We'll be talking more about how we're expanding that collaboration. And that gives us a lot of data on approved drugs and on over 1,400 different known mechanism of actions that are inside of these cancer drugs. And so that gives us the ability to then replicate some of these MOAs and look at the genes in different drug types. One of those that we highlighted, and I'll talk about how we went from the in silico to the real world, was that we looked at a very specific mechanism of action for 184. In that, we use machine learning models to rank the gene expression importance of the various important genes. Various models came up with various signatures ranging from 20 to 9 genes. And we looked at the high rank feature, and we really deciphered that PTGR1 in this case really required further experimentation and in-depth analysis as the driving gene required for success of this drug. And so just put that in the back of your mind. So this is a long process. We used a lot of different models to do it. We went back to the lab to test it. We've been tuning the hyperparameters, both in cell lines and PDX models with biological relevance, without biological relevance, looking at how the models perform to see under what conditions they perform best, and then actually having that perfect or ideal model. That ideal model, once we have that, we can create a gene signature panel for a companion diagnostic, and we can apply it to the cancer patient population to screen those people who fit that signature for our trials. That signature can become a companion diagnostic, and we can use that for patient stratification and guidance for clinical trial design and studies. So how do we do that? As we get various data and expression data, we run 2 different groups, training and testing data, typically 70-30 or 60-40. We have over 13 different high-level models, algorithms, both based on regression and classification that we test. These go through various levels of hyperparameter tuning and tracking. These have now generated about 78 different registered models that we have in our system. This will continue to grow rapidly. And these are all managed through MLflow. The models that have the best performance, either in correlation, area under the curve, prediction as tested in the actual lab, these are then annotated. These models are now aware where they do best and how they do best. So as we generate the superior drug models, we continue to tune them, we continue to add values to them, and all this is automated. All the metrics around performance, the metrics around tuning, and the metrics around which data sets, all this is containerized and stored in the cloud. And so these models continue to get better and better, and they allow us to better understand the mechanism and what is required for the drug and the tumor to have the kind of effect, meaning real change in patient care. So what does that mean? Typically, our process when we first started, it took us a few days to select the data, it took us months to reduce and tune it, and then finally upload the files. Today, we've automated that. It takes us less than a day to select the data, sometimes hours. It takes us now weeks or even days to do all of the automated feature reduction and ML tuning. And it takes us less than a month or a few weeks now to automatically now upload, track, improve, link the models. And so that is that's hundreds of thousands of dollars in drug development. We're taking something that used to take months to develop and do these pipelines now down to weeks. And now this only gets more complex because, again, we went from 3 drug programs last year to now over 8 drug programs. And so we've developed and integrated a machine learning operations environment called MLflow. We use this to find the best performing models and various metrics by sorting the columns, by looking at the experiment IDs, by looking at the end values. And then more importantly, we can look at the information on any one of these models. This allows us to look at the comparative performance during the training and testing environment, look at which model performed best and why, based on things like Pearson correlation or area under the curve or various RMSE values. Now, this is a summary that you're looking at from nearly 1.9 million models that we ran for one of our drugs. This is important because PTGR1 came up as one of the most important, but very importantly, the subgenes that came up were also related to a fundamental feature of the drug, meaning that it worked when NER genes were deficient or HR deficiency was seen. So deficiency in the DNA damage repair, which is an important value for why this drug works. So again, we automated this, took 13 methods, over 119,000 models that were generated and trained in one day. This is a massive number of models that were all tracked, all containerized. And as the pipeline runs in the cloud, we can do this faster and faster and faster. Now we can do this in hours that then go into weeks to run all the various different models. And now we can know where certain algorithms work better or worse, and under what conditions, and we can tie those to very specific drug development questions. All this has been done internally. There are commercial platforms that allow you to do this type of work. They cost hundreds of thousands of dollars, and they're not really generated for drug development and not for oncology. So we believe we have now really the best-in-class engine for specific development, managing, and tuning algorithms for various drug development questions. Let me give you one specific example. As I mentioned earlier, PTGR1 was validated to be involved as the mechanism of choice. So what happens now in the lab? If you look at the blue and gray curves, those are cells that actually have high levels of PTGR1. This was now put into a CRISPR experiment in the lab. The orange and yellow lines were the same cell lines, but PTGR1 was suppressed. So PTGR1 could not have the expression. And so if you look at the cell lines, they really didn't respond to the drug, very flat. Whereas the cell lines that actually did have overexpression of PTGR1, which was hypothesized to play the driving role, you can see that the cells died very, very quickly at about 200 nanograms per ml, very low dosage, nanomolar. The cells are largely dead. This is almost a binary event. This is a 1-0. Very rarely do you get this. And again, all of the ML models pointed in this direction. And as we looked and did the biological feature selection in the model, it really helped us tighten it up and confirm the strict dependency of our drug to PTGR1 expression. And that understanding the mechanism allows us to now go pick certain areas where PTGR1 is overexpressed. One example is certain GBM. Again, if you look at how this drug works, and this is in actual mice, you see complete regression in one of the models. This is an actual living mice. And in the other mice that had neurospheres from patients, you saw 3 out of the 4 mice have no tumor growth after final dosing. And I do have to say this is without actually fully optimized dosing. And so you see that the thesis that we had in silico played out in gene editing experiments, continues to play out in vivo in mice as the driving. And more importantly, we see massive regression. This is something you don't see in many GBM cancers. Not only are we lucky to see it in GBM cancer, but we did it also in pancreatic cancer in mice, where this gene was highly overexpressed, PTGR1, but also, very importantly, had another feature. It had BRCA deficiency. And so it also had what we call the DNA damage repair deficiency. And so as our model suggested, when you have both, you get a massive response. And that's exactly what you see, the difference in the blue and green line. You saw the green mice, that set of mice, basically cancer went away, whereas the ones that were untreated, the cancer grew over 50-fold. And again, this is after sacrificing the mice. You can see the average volume in the ones that were not given the drug as about 587 or 600 cubic millimeters, whereas the ones that were given the drug, you can see the cancer almost went away entirely. And in one of, you had no tumor left at all. So again, it's in silico that now is actually happening in real world. And we're doing that at a fraction of the cost. By understanding the mechanism of action, we can use pathway and gene interaction next to identify potential combinations, to identify where the mechanism may or may not be present in various types of cancers, and select other cancers. That's how we picked ATRT. So once we understood that, we're able to look at what is now associated with driving that kind of activity. And we looked at ATRT, another highly malignant brain tumor. We didn't have this program when we went public, but we did several months after because we knew that chromatin remodeling was implicated in some of this DNA repair. And so, as I mentioned, DNA repair is a key feature in that gene signature. And again, of course, if you look at the very bottom slide, you can see that that's in real tumors going from in silico prediction to real tumors. The tumors that were not given the drug grew. The tumors that were given the drug almost went to zero. We're now in a collaboration with Johns Hopkins with this drug, and we plan on being in humans next year. So understanding this allows us then to take these algorithms and models and look at new drugs. This is how we generated a totally new drug, de novo, called 284, that had similar metabolic activators that we saw in the oxidoreductase class of 184. And now we see that this drug works really well actually in blood cancers. We'll be presenting at ASH. We just put a poster up on that very recently. And so drug synergy is a big thing that we look at now because the drug synergy gives us ideas about how drugs will work together. And we're able to now do various models that show how drugs work together. One of those suggestions was an FDA-approved drug for treating hypertension because it downregulated the expression of a key DNA damage repair gene called ERCC3. We found that in models that when ERCC3 was deficient, the tumor became highly sensitive to the drug. And so we said, well, let's take this to the lab. We took it to the lab and we administered spironolactane, which was an ERCC downregulator with our drug. You can see it became massively sensitive. And so again, this is the kind of real world implications. So we go from in silico hypotheses to driving ML feature selection to understanding what are the driving genes and then validating that in the lab. And so again, this allows us to create a model of connectivity, and the models of connectivity allow us to look at various types of cancer drugs and see how those can be combined with our drugs and perhaps boost the efficacy above the current standard of care so that we can get closer and closer to really controlling cancers and really potentially curing them. And so these are problems that are worth hundreds of millions or billions of dollars to pharma. In our own case, we took one of our drugs, combined it with an antibody, and we have some initial data, again, from the lab to support the fact that a combination of our LP-184 type drug with an antibody has massive effect on improving the cancer kill rates. And we've seen that in line with other FDA-approved drugs, as you see on the left-hand side, but also in a highly challenging refractory leukemia. You see that in the blue within 4 hours with about, again, 100 to 200 nanograms of our drug combined with the antibody to really hit the cells, that we got massive response. And not only massive, but really 100% response in 4 hours of cell culture, which typically you see with other drugs about 30 to 50% response. So again, we're seeing very good outcomes in our own programs that support the use of these kinds of data-driven methodologies to make fundamental decisions about the drug, about the mechanism, and very importantly, about how drugs can be brought together and what subtypes of cancer, because there are literally hundreds and thousands of types of cancer. So we're developing what I believe is the world's most powerful and largest end-to-end platform for oncology drug development. We're very open to collaborations with others. We have collaboration, as I mentioned, underway with Actuate, which is going very well. And we probably will have, we're looking to do several others now. So with that, I'll take time out. I know I'm going up against my time limit and open it up to any questions that people may have.
Thank you so much for that, Panna. Very exciting stuff. We do not have any audience questions as of this moment, but anybody who does have questions for Panna about the truly exciting stuff that that Lantern Pharma is doing to transform these potentials into actualities, please feel free to pop those into the Q&A box, and we'll do our best to address them during our roundtable later. And just FYI, for anybody who's interested in seeing sort of a larger scope evolution of the stuff that Lantern has been doing. There are archived recordings of some previous webinars that Deep Pharma Intelligence has done. I know that Panna was here with us in our December 2020 webinar some ages ago. So feel free to check that out.
The platform's come a long way since then.
It's very clear, indeed.
Yeah, it's come a long way. So and it continues to evolve pretty rapidly as we've automated various features, and automating the algorithms and the ability for the algorithms to start now training and tuning themselves has taken our ability to a whole nother level. And so and now we're improving in certain rare cancers, and now we're also adding now immuno-oncology data into it to be able to predict that. So we expect that by next year we'll be north of 25 billion points, and probably somewhere in the range of 300 to 400 different algorithms that are able to address various problems.
Well, that's really fantastic. AI is the quintessential accelerative meta technology, right? And it's pretty clear that that's playing out in practice here. So thank you once again, Panna.